Good afternoon. It's a great pleasure for me to introduce today's world speaker, Dr. Karen Duff. She's a professor of pathology and cell biology at Columbia University Medical Center and at Tau Institute for Alzheimer's Disease Research. After getting her PhD in molecular genetics from the University of Cambridge in the UK, in the lab of Dr. Sidney Brenner, she did a few years of postdoctoral training with Dr. John Hardy in the Department of Biochemistry and Molecular Genetics at Imperial College in London. And then she moved with the lab to the Department of Psychiatry at the University of South Florida, where she was promoted to the rank of assistant professor in 1993. Then she moved as an associate professor to the Mayo Clinic in Florida in 1996, and then to the University of New York in 1998. In 2006, she progressed to her current position of professor at Tau Institute and professor of pathology and cell biology at Columbia University with a tenured appointment. Dr. Duff has over 100 publications in prestigious international journals, sits on 3 editorial boards, and has received numerous awards in recognition of her scientific achievements, including the Potemkin Prize from the American Academy of Neurology, which is one of the most prestigious in the field of Alzheimer's research. Her research is mainly focused on Alzheimer's disease and the contribution of tangles to disease, and particularly the role of aberrant phosphorylation of specific proteins, such as tau, in the progression of the disease. Today, Dr. Duff will present data supporting the targeting of both amyloid and tau to reverse neurodegeneration and cognitive impairment in Alzheimer's disease, and will discuss several therapeutic approaches, including kinase inhibition and modulation of autophagy, that aim to reduce pathological forms of proteins in the Alzheimer's brain. Finally, she will also present exciting data on a new mouse model her group created to study the spread of the pathology. So without taking any further time, let me welcome Dr. Duff. Thank you. Sorry, there will be a reception in the library after the question and answers. Please come to the reception afterwards if you like. Thank you.
Thank you very much, Francesca, and thank you for the nomination to this highly prestigious speaker series. The alumni for this series are really quite overwhelming. I noticed that of the Alzheimer people, you've had Karen Chow-Ash and Mike Hutton come to talk to you about Alzheimer's, and they represent more of the amyloid pathology with Karen Chow and the tau pathology with Mike Hutton. So I thought it would be quite good if I sort of introduced you to more of the concepts that where we're at now with the field in terms of how we're reconciling the contributions of amyloid and tau, and perhaps what we need to learn from the clinical trial data, the limited clinical trial data we've got so far, and how we should maybe be thinking more about this disease from now on. So just to orientate you, I realize mixed audience, just to orientate you, the pathological features of Alzheimer's disease include 2 major lesions in the brain: the extracellular deposits composed of the A-beta peptide. Am I using it? Oh, I'm using this. Okay. Composed of the A-beta peptide, which develops in regions involved in memory and in the cortex of the brain too. Intracellular neurofibrillary tangle, shown here, composed of abnormal protein tau, which in Alzheimer's disease takes on a particular conformation, the paired helical filament conformation. There's an inflammatory response that occurs concomitant with these lesions, and that includes activated astrocytes, astrocytes and microglia that tend to surround the plaque lesions, a very close association with the plaque lesions. There's also an immune response in terms of complement pathway activated, and all of these features occur in the context of loss of synapses and the development of abnormal cell processes and degeneration of neurons. And the ultimate sequelae of this is the cognitive impairment that's characteristic of Alzheimer's. So the main idea in Alzheimer's disease was that the amyloid plaques are composed of a peptide called A-beta, and A-beta is produced from a type 1 transmembrane protein called the amyloid precursor protein, or APP for short. And the APP was considered very fundamental in the etiology of Alzheimer's disease for a number of reasons. Firstly, APP is on chromosome 21, and triplication of chromosome 21 leads to Down syndrome, and Alzheimer's is a fairly invariant feature of Down syndrome. And secondly, the APP precursor protein produces a peptide called A-beta, and A-beta is forms the core of these amyloid plaques. And the pathway by which it's produced is shown here. This is the APP protein, and through a series of enzymatic cuts, you can produce either the A-beta peptide, which is between 40 and 42 amino acids long, or a shorter peptide if this cleavage occurs before this cleavage. So you can generate this A-beta peptide through a number of cleavage events, and that A-beta monomer can go off and aggregate first into dimers, trimers, and ultimately oligomers, and from there it can form into multimeric structures that form the structure of the senile plaque. So this very much implicated APP processing in Alzheimer's disease, but what really sealed its fate, and the reason why APP and A-beta production is the major target for drug companies and really has been the driving force behind decades of research, was the identification of mutations that occur in the APP molecule that are associated with, that actually cause Alzheimer's disease. And those mutations can occur either at the N-terminus of the molecule, often affecting the cleavage of the base enzyme, or at the C-terminal site affecting the cleavage of the gamma secretase. A number of other genes other than APP have been implicated in Alzheimer's, and actually this C-terminus cluster and this gamma secretase was actually found to be the second genetic cause of Alzheimer's disease in that it was found to be the presenilin protein, which is part of the gamma secretase complex, and these proteins all regulate the production of A-beta, or the length of the A-beta that's produced. So a number of issues actually suggested that APP processing was the major target, that should be the major target therapeutically, and this has driven the field for many years. So what happens once A-beta is produced? And you'll see I use the word A-beta and amyloid. I often put them together. And this reflects some of the shift in the rationalization for how Alzheimer's disease is caused. We used to observe amyloid plaques in the brain and try and correlate downstream effects with amyloid plaque formation, but it's become clear that the A-beta peptide, or some oligomeric form of it, is perhaps more responsible for the downstream neurotoxicity than the actual amyloid plaque. So although the amyloid cascade hypothesis, as it was termed by John Hardy and his colleagues a long time ago, although it still refers to amyloid, the amyloid plaque is probably not the neurotoxic lesion, although it definitely contributes to the neurotoxicity. It's probably some intermediate form of APP, such as an oligomeric or dimeric form of A-beta. So the idea here is that A-beta generation, either through mutations or in sporadic disease, which is the 95% of cases are sporadic rather than familial, A-beta can produce a number of effects, including oxidative stress, excitotoxicity. It aggregates into the plaque lesions, which have a negative effect on the brain in their own right. It evokes an inflammatory response. And importantly, it leads to some effect on tau, which leads to hyperphosphorylation of tau, which is associated with the formation of these neurofibrillary tangles, the secondary lesion of Alzheimer's. And Alzheimer's is pathonomically defined as a disease of plaques and tangles. So you get into a little bit of an issue if you try and say someone has Alzheimer's disease, but they don't have these two lesions. However, there is a whole subset of Alzheimer's type dementias that only have either plaques or tangles. So downstream of this is, of course, cognitive and behavioral abnormalities and cell death. And so the whole cascade has been related back to A-beta. But I'll go into a little bit where I think this falls down, this whole cascade hypothesis. So I just wanted to show you quickly, and someone had asked me about this model. This is when you make a transgenic to model this disease, classically what we've done is we've taken mutations in an APP gene or in a presenilin gene, and we put them into a mouse, and at high enough levels of expression, you can recreate the pathology of Alzheimer's disease. Here you'll see the plaque pathology, and using specific antibodies, you can see A-beta forms that are represented in the human AD brain. you get degenerative structures, you get silver positive, you get thioflavin S positive plaques. And around these, you'll get your microglial response and your microglial and your astrocytic response. And you also get these little dystrophic neurites. This is tau protein staining on the little dystrophic neurites that pop up around the plaques. So we've been able to model the pathology pretty well in mouse models. They were classically done in mouse models for many years. This was the first of the rat models that was produced to model the disease. And in addition to the pathology, we also get, of course, the classic degenerative phenotype. And here you can see an LTP deficit. This is the transgenic, and this is the normal. And you can see a deficit in LTP, and you can see a deficit in performance in the water maze. And this is sort of a classic water maze test for memory and learning. And I won't go through this. I'm sure you're pretty familiar with these type of pictures. The ability of the mouse to learn where a platform is using cues around the room as an aid to memory so that it can remember where the platform is. So you see this is represented in both the mouse models and also in this rat model, suggesting that there is an amyloid or a beta-related deficit in both LTP and in memory as the pathology burden increases and as the animals age. So moving on a little bit from amyloid, it's been discussed a lot, and I would rather concentrate more on tau because I think this is more where the field is moving to. So to just give you some background information about tau and tangles, this is a brain with Alzheimer's disease, and you see the classic tau lesions, and these are the neurofibrillary tangles that are intracellular lesions where this tau takes an abnormal form and accumulates in the cells. And then these dystrophic neurites, which are here, they are on the end of the processes, and these swell up and accumulate hyperphosphorylated forms of tau, and they tend to circle around the amyloid plaques. In addition, you get neuropil threads. I think there's one there, which is probably a little bit of a dendrite that has some tau accumulated in it. So what is tau and why is it involved in degeneration and development of pathology? Well, tau is normally a microtubule binding protein, and here's a microtubule network, and here's the tau in blue, nicely bound to the microtubule, stabilizing the microtubule. And in the disease condition, what we see, and we don't know the reason why we see this, but what we see is a degeneration of the microtubule network. It starts to disintegrate. Tau has come off the microtubules, and it's forming aggregates out in the cytosol. So it's no longer firmly associated with microtubules, it's now free in the cytosol. And along with this, we see a redistribution of tau from the axons into the cell bodies, into the somatodendritic compartment, the cell bodies and the dendrites. And this is usually the very first indication that something's gone wrong with the cell and it started to form a tangle. So the tau protein is an interesting protein, and in terms of modeling it, in terms of trying to recreate pathology, there's a few issues with it. The human form of tau has 6 different isoforms, and they differ in 2 positions. Here, there are 3 microtubule binding domains in 3 of the isoforms, and in 3 of the isoforms, there's 4 microtubule binding domains, and these tend to increase the stability of the tau in its relationship with microtubules. There's also differences here at the N-terminus, and the differences between mouse and human is that mouse only have this 4-repeat form, these 3 4-repeat isoforms, whereas humans have all 6. And this difference in isoform ratio, although it doesn't cause tauopathy in mice, it has significant implications for humans. Several of the diseases with which tau is aberrant, and there's about 30 different dementias in which tauopathy is a part of the phenotype, obviously including Alzheimer's disease as one of them. In some of these diseases, you actually get a shift in this isoform ratio. You can get more 4-repeat forms in PSP and CBD, or more 3-repeat forms in Pick's disease. Most of them have variable contributions, and Alzheimer's disease itself, we're not sure what the impact of different ratios of the repeats are. We're not even sure if it changes with disease, but usually it's a 1-to-1 ratio in Alzheimer's disease. So quite a significant impact of these different isoform ratios. And in terms of the genetics of how this can happen, Alzheimer's disease doesn't show really any mutations in tau. You'll remember I told you mutations in APP can cause Alzheimer's disease, but mutations in tau have not been identified. A haplotype variant has been identified, which increases the level of all the tau forms, but we haven't seen any mutations in tau causing Alzheimer's. We have, however, seen mutations in tau causing other dementias of which where tangles form, and so we know it can be a primary mutant, a primary pathology-causing protein in its own right. So for the modeling, we took some. There's two different types of mouse model for the tauopathies. The first mouse model was a mutant tau mouse model, which overexpressed 4-repeat tau. But in terms of Alzheimer's disease, the best mouse model to replicate the human condition is this mouse model that I created and gave to Peter Davis, and he worked up some of the pathology. And this one takes the whole tau gene with the human tau promoter, and it's a normal wild-type gene, and it puts it into a mouse, and we put it onto a tau knockout background, and it's a very nice model making all 6 isoforms of human wild-type mouse without mouse tau. And the phenotype is what you would like to see in a mouse model. It's a progressive phenotype, so at 6 weeks of age, you see what would be considered the normal distribution of tau. This is all axonal. You don't see any cell body or dendritic pathology. But as the mice age, you start to see a shift, a relocation of tau into the cell bodies in areas of the cortex and the hippocampus. And by 13 months of age, you've got a pretty severe accumulation of tau in areas where it was not previously located. The tangles themselves, this is what you would see on these models, and this is the sort of thing we're looking at when we're trying to define whether a mouse model has pathology, and then if we're giving it a therapeutic, whether it's correcting it. These are the tangles that you would see in the mouse model, and I use tangles loosely at that point. Tangles are usually poorly defined when people are talking about them. A pretangle is a form of tau where phosphorylated tau is accumulated in cell bodies like this, and you see this staining in the cell body. What you can go on to is then a full-blown mature tangle where you get conformationally abnormal tau accumulating, and it can be positive with thioflavin S or a marker of a conformational abnormality. Once it's gone to this form of tau, then you start to see by EM, you see tau in filaments. Normally it's a soluble protein, but as it aggregates, it has formed filaments, and they have a particular periodicity as they form these paired helical filaments if you've got a valid model of Alzheimer's disease tangles. That periodicity actually differs between the different types of tauopathy. If you have a mutation, you actually often have a different formation of the filaments, which is quite interesting. So we can generate mouse models that have progressive pathology, and they form very good features of the disease. And in a recent publication that we had, we were able to show that the mouse models, this wild-type humanized mouse model, actually had a cognitive deficit as it aged. At young ages, it didn't have a cognitive deficit. At older ages, it did have a cognitive deficit, and that was shown in the Morris water maze. In addition, there's an alteration in LTP. It's a particular type of LTP, a high-frequency LTP, and it tends to suggest that there's actually a presynaptic deficiency, and this is associated with increasing tauopathy. So we have a nice, valid mouse model for tauopathy. And as I said, in addition, we have some of the mutant tau mouse models that form a very accelerated form of tauopathy, which replicates nicely the frontal temporal lobe dementias, Pick's disease, CBD, etc. So now I'm going to come on to, so that's the background of how we've generated our resources for this disease and some of the motivations. So this is a little bit more controversial. So what I'm going to do now is I'm going to discuss where we're at with understanding our disease, because it has great impact on how we try and deal with it therapeutically. And some of the feedback that we're getting from the very limited clinical trials that have been done suggests that we do have a problem with how we're actually dealing with this disease, and I'll come back to that in a minute. So this is this idea that there's maybe two different forms of the disease was embodied in a neuron perspective piece by my colleagues Scott Small and I at Cambridge. Not Cambridge, I was in Cambridge on Monday, sorry, at Columbia. And so what we sort of are thinking is that the model that's been proposed, where A beta is the driver, and increasing A beta leads to an increase in tau abnormalities, synaptic loss, and cell loss. That's fine for an early onset familial form of Alzheimer's disease, such as that generated by APP mutations, presenilin mutations, and that this is fine. This is fine for this model. However, most of the cases of Alzheimer's disease are late onset and sporadic in nature. And we're concerned that by applying this serial model for all models of Alzheimer's disease, we're actually missing something, and this will have some impact in terms of how therapeutics respond. So the model that we propose for sporadic AD is one of two things. You could have a dual pathway model, and in this model, what this means is that you've got pathology, you've got both pathological events occurring. You've got an increase in A beta, and also an increase in something to do with tau, and we've labeled it tau hyperphosphorylation because that's sort of our starting point for the tauopathies. But the understanding here is that these two pathways are set off in parallel. And they're caused by some dysfunction upstream, and we don't know what that could be. It could be advanced age or some kind of sensitivity in a particular region to some insult. And some of those insults could be risk factors, such as genetic risk factors, ApoE4, diabetes, cardiovascular events, stroke, brain injury. These are all known risk factors for AD. And so what we're proposing here is that you have an upstream dysfunction and that these set off both pathways. And the downstream effect is synaptic and cell loss. Where this becomes a problem is if you start to reduce A beta with an anti-A beta therapeutic, you're going to remain, this part of the pathway is going to remain unaffected because basically it's being set off in parallel. The second part of this is actually that you do get both of these pathways, but most of it's driven by A beta. And this is more similar to the serial model, where you can increase A beta, that has a knock-on effect to tau, etc., and downstream effects. And this might mean that if you can reduce A beta to some extent, but you'll still have a bit of a problem coming in here, but if you do it early enough, this should be reversible. And so understanding your model is, I think, very important for monitoring the effect of the therapeutics. So the idea here would be that A beta enhancing mutations in APP suggest that it accumulates and drives the downstream pathology, but the opposite would be that you cannot actually prevent this from happening just by preventing A beta from accumulating. Sorry, I'm missing a slide. So is this something we should be concerned about when we're looking at therapeutics? So I didn't want to go through too much of the therapeutic approaches that have been tried in Alzheimer's disease in the clinic. There were many, many of them, but I wanted to just go through some of the most robust data we have to feedback on whether we're dealing with something here is from the immunization or immunotherapy approaches for Alzheimer's disease. And this approach that's been used clinically in a number of forms comes from data from a mouse model where they used immunotherapy to reverse the pathology in the mouse model with amyloid plaques, and to also reverse the cognitive impairment. And the model was, this approach was developed by Dale Schenk. It was suggested by Dale Schenk and has been applied by Elan Pharmaceuticals. And there have been a couple of approaches, one where they inject peptide to A beta. That raises an immune response, and that is correlated with an improvement in measures of pathology. And the second is in passive immunization, where they actually inject the antibodies themselves. And both approaches have been very effective in the mouse models, and both approaches have gone into clinical trials now. So what happens when you look at the results of the patients on these clinical trials? So unfortunately, the first trial with the active form of the immunization was halted due to encephalitis occurring in the patients. And so we don't have very much data back from those trials. But what we do have shows that whilst the patients were on the trial, they did not improve cognitively very much. There was a small improvement on some measures of cognition that had to be sort of adjusted and worked up so that they showed a post hoc difference in cognitive decline. What I think is interesting is what happened here. So this is a pathology report on patients who'd come to autopsy about 6 years after the trial had been stopped. So these were patients who'd received a couple of doses of the drug and had been monitored whilst the early days, and then when they died, the pathologist had access to their brains. And there's only a handful of patients in this study. A second study has been performed on more patients, and it essentially looks similar. And what you're looking at here is this is the mini-mental status of the patients, and this is a measure of cognitive performance. It goes from 30 to 0, with 30 being unimpaired and 0 being highly impaired. And this is the baseline mini-mental score. Obviously, you could take it of the patients before they went on the drug. And the thing to note here is that these two patients were pretty high functioning. A mini-mental of 25 is in the range that we call mild cognitively impaired. It's barely Alzheimer's disease at all. So these were pretty unimpaired patients. The dose of the vaccine did elicit a reasonable antibody titer, so we know that there was an antibody titer raised in these patients. And then at autopsy, the pathologist, James Nichols, was able to monitor how much plaque amyloid he thought had been removed from the brain by this immunotherapy approach. And that's a bit of a difficult question, because you can imagine you don't have pathology, so how do you know how much was there before? Well, he used sort of scarring and sort of clues that there was an amyloid plaque that had been resolved to some extent. And what happened was in 6 of the 8 patients, there was quite a considerable removal of amyloid plaques, and in 2 of them it was very extensive. The thing to note here is the BRAC stage of the patients at pathology. BRAC staging is a classification of Tangle load, and it's sort of a measure of how much Alzheimer's disease you have. Low BRAC numbers of 1 to 2 are very early Alzheimer's, 3 to 4 are intermediate, and 5 to 6 are severe, with 6 being the most. And you can see that these patients had very, very severe pathology, neurofibrillary pathology at end stage when they came to autopsy. And in addition, they were completely demented with many mental scores of 0. So this was intriguing, because it suggested that this drug approach had actually hit target, which is the first thing to note. We've had real difficulties getting A-beta reduced in the brains of patients, but we know that this antibody had actually reduced amyloid, but we had not protected the patients from disease. The tau pathology is particularly interesting, and that's why I'm bringing this to your notice, and this is actually what's driven a lot of our work now. As I mentioned, the BRAC staging goes from 1 to 6. Stage 4 is about what you would have with a mini-mental stage of this. Stage 4 is sort of intermediate, goes to stage 5, stage 6. And one of the critical things with BRAC is not only how much tangle pathology you have, but where it is in the brain. It tends to spread out of the initial areas, and it goes into new areas of the brain. I'll show you a diagram of that later. So because they probably started at an early BRAC stage and ended at this BRAC stage, we actually think that the pathology had spread into new areas, despite the reduction and removal of A-beta as the patients were on the therapy. So should we interpret this negatively? Should we say that, yes, A-beta and amyloid is not the target because these patients didn't show much of a cognitive improvement, if any, and secondly, they still died of full-blown Alzheimer's disease, even when they had reduced levels of amyloid in the brain? Well, that could be one interpretation, but there's also some caveats to this. So it's possible that we got the wrong type of A-beta. You'll remember I told you that there's different forms of A-beta, monomeric, oligomeric, dodecameric, fibrillar, and maybe we removed the wrong type with the immunotherapy. Maybe the dose was wrong, the trial was too short, number of samples too low, or the drug was given too late in the disease, and those are all valid criticisms. It's also possible that 6 years after the trial ended, the pathology had a chance to reactivate the tau pathology, and that in fact it would have stayed down and gone away if we'd continued the trial. But the most important one is probably that the whole immunotherapy was administered to the patients when they were too far advanced in their disease. And the reason why I pointed out those high mini-mental scoring patients was that stage, that MCI, mild cognitive impairment stage, is about as earliest as we can intervene in Alzheimer's disease. It's the first time patients come to clinic. You really, without better biomarkers, we can't get much earlier than that. So my concern with this is that although we often administer these drugs in severe or moderate Alzheimer's, even earliest interventions may not be that effective. We do know that even those earlier stages have a very high amyloid and tangle load, almost as high as they'll ever get in Alzheimer's disease. So my big concern is that with our current therapies and our current state of knowledge about biomarkers, we are not going to be able to intervene early enough. At least if we're going to go for an anti-A beta approach, we're not going to be able to intervene early enough to knock down the tauopathy. So do tangles accumulate and propagate even if A beta is reduced? Well, this has become a part of the field that I think is very concerning. And here's some data on an inducible mouse model. This is a mutant tau transgene under the control of a TET activator, which is driven by the CAM kinase promoter. And you can turn the tau transgene on and off. And if you have it on and turn it off a couple of months later, you can resolve all of the tau, mutant tau in the mouse. However, if you have it on and turn it off after a certain stage, not only can you not remove the tau, but in fact it accumulates and propagates. It draws in normal mouse tau, and the tangle pathology becomes worsened. It seems that the stage when this little band here, this is a 64 kilodalton form of tau, which is an insoluble tau form, it seems that once that has been reached and achieved, then the pathology is not reversible. At least not by switching down tau levels. And so this is a big concern, that tau pathology can propagate in the brain. This has been backed up by some new work that we're really entering into the field. The field is entering into a whole new era of looking at pathology spread, and this mimics what's going on in other neurodegenerative diseases. And we're taking a lot of our cues from the prion field. And so some of the data is starting to suggest that propagation of tau tau misfolding can occur when you add aggregates outside. They can be internalized and they can sequester the normal tau and turn it into an aberrant form. A very interesting experiment done by the French group of Marcus Tolnay injected lysate from a mouse that had aggregated tau into the brain of a mouse that had normal tau in a particular area. And they were able, after about a year of incubating the mice, to see pathology developing, not only in the areas around where the injection was, but in areas that were distant to it, that were quite considerably far away. And this suggested very strongly that, tauopathy could spread throughout the brain from an initial site of injection. So we're very interested in this idea, and we've started to look into how to model this, and I think this opens up a whole new area potentially for therapeutic intervention, because a lot of people have tangles and tau in the brain. You don't have to have Alzheimer's disease, and you can have quite a high burden of tangles, also of amyloid. There's something about where it is and the density of the lesions, and then where it goes to. So the staging of Alzheimer's disease, as I said, this is the BRAC staging. And in the earlier stages, it's here in the entorhinal cortex. The tauopathy is really fairly concentrated in a particular cell type called stellate neurons in the entorhinal cortex. And as pathology progresses, as the disease starts to worsen, the pathology spreads out from the entorhinal cortex into the hippocampus, and from the hippocampus into the neocortical areas. And this is very, very severe Alzheimer's disease. This is moderate. This is probably pre-Alzheimer's, prodromal Alzheimer's disease. So we've really got a defined spread of pathology from an initially vulnerable population into the rest of the brain. So if you can intervene maybe at this stage, or certainly at this stage, you can probably prevent Alzheimer's disease, even though you'll have tangles in the brain to some extent. How is this happening? Well, this is a big question. We don't know. And these are some of the things that we're trying to model and look at. How can tau pathology spread? So one of three possible approaches. Perhaps it's cell autonomous. Maybe dysfunction in cell A in the entorhinal cortex induces dysfunction in neighboring cell B, induces dysfunction in neighboring cell C. So this is a spread of dysfunction rather than the pathological molecule itself. Maybe the molecule, for example, misfolded tau is secreted from cell A, and that induces a local area of dysfunction, which is spread on, picked up by cell B, and that it produces a wave of dysfunction. Or possibly, and this is very similar to what you see in prion, is the secretion or transfer of a molecule from cell A is actually picked up by cell B. You get an abnormal tau conformation, it secretes its molecule, it's picked up by cell C. So there's several possible modes of spread of this pathology. And we wanted to start to look at this in a number of ways. And I do apologize now, we didn't get quite as far in the data as I had hoped. So I can start to show you some of the data. But I wanted to introduce this because I think this is a new concept in tauopathies, which is also of relevance to synucleinopathies and any of the lesion-forming diseases, is this spread of pathology. So I sort of want to give you some ideas of how we're doing this. So what we're doing is we want to generate pathology in the entorhinal cortex. And we're lucky because Mark Mayford's group had identified a promoter from the neuropsin gene that's really regionally restricted to just a couple of layers in the entorhinal cortex and the para and presubiculum. And it's expressed, it drives the expression of the TET activator. And in this experiment, he drove expression of the LAC-Z gene as a reporter gene. But what we want to do is we want to drive expression of a tau transgene, and we're using the mutant tau transgene that's in this mouse. This is the one of Karen Chow's tau mice, and it has, when it's driven by the CAM kinase promoter, TTA, it has very severe and robust pathology. So we're crossing a neuropsin promoter mouse, TTA activator, with the tau mutant responder mouse. And this is the sort of thing you see in the neuropsin TTA tau mouse, and this is staining for both human tau and for H&E staining. And this is the entorhinal cortex. You get expression both in the medial entorhinal cortex and in the lateral entorhinal cortex, some in the parasubiculum and presubiculum, but that's about it. You really don't get expression in other areas. So this is really nice for doing mapping studies. And what we're going to do here is we're going to use the distribution of pathology to actually study where the tau is spreading through just to its projection areas. And in this model, the projection areas here, you can see the projection areas actually here. Part of the dentate gyrus, you can see the middle middle layer of the molecular layer of the dentate gyrus is a projection area from the entorhinal cortex. And you see tau has distributed from here down to here, and it's just going through one axon. What we're really interested in is, does it cross a synapse from these cells into other cells? And from there, does it cross from other areas of the hippocampus to areas that start to be affected later in the disease? And if they start to spread up here, these areas are not related unless they're through a synapse to areas down here. And this is then transsynaptic spread. So we're using this sort of approach to try and map exactly where we see pathology developing. Now, we're not all about pathology. We've learned our lesson that perhaps pathological lesions aren't actually the disease. Maybe the Tangle pathology or amyloid plaques are not the disease. Maybe you're looking at an intermediate, an A-beta form or a tau form that you don't see in a Tangle. Also, maybe the disease is not all about lesions at all, or even intermediates. Maybe it's actually just cellular dysfunction, something, glucose metabolism, or something that you can pick up in other ways, and that the pathology is just one aspect of this. So we're coupling these mapping studies with studies using fMRI to look at dysfunction of regions, the entorhinal cortex, and of regions that are associated with them by transsynaptic connectivity, and starting to map where this dysfunction is. And in that way, we're hoping to actually to answer part of the question about, is it dysfunction that spreads, or is it pathology, or those sort of answers. And we have some preliminary data, but I won't show you. It's really too preliminary to bring it up. But we can see dysfunction down here by fMRI. We have not aged the mice above 4 or 6 months to be able to start to see it in distal regions. But we think that this approach should be informative. What we are able to start to do is to start to look at different epitopes of our molecules of interest and see if the pattern of distribution, the distribution of them is the same or different, and what it is about those different epitopes that perhaps is giving us a different pattern. So here again, this is the entorhinal cortex where this is expressed. And we see this is an antibody called MC1, which is a conformationally abnormal form of tau. It's a human-specific. Here's the control showing no staining. It's an abnormal conformation. You can see it's in the projection area. So this form of tau just flows down to the terminals of the axons. In comparison, a phosphoepitope of tau, this is CP13, which recognizes phosphoserine 202 and threonine 205, This has really quite a considerable different pattern. You see a little bit in the projection areas. Here's the cell body. Now it's in the cell bodies. Here it was not in the cell bodies. And you see it starting to appear up here in the hippocampal fields, in the cell bodies of the pyramidal layer of the hippocampal fields. We also think, but we haven't had old enough mice really, we think that we're seeing something starting to occur out here in other regions of the cortex. And this sort of pattern it would be very, very nice to show, if it's confirmed in older mouse and it gets worse, this would be exactly what we'd expect if you've got some kind of spreader pathology. This would be very exciting if this was human tau, because then that would tell you that it's spread of a molecule transsynaptically. Unfortunately, we don't have phosphoantibodies that recognize only human tau. So this recognizes both mouse and human, and this is the control mouse. You can see that some of the mouse tau is recognized by this antibody. So we need a lot more antibodies to be able to do these sort of studies and start to map together what's going on in terms of the distribution of tauopathy and tau forms. Just as a comparison, we've done exactly the same thing with an inducible APP transgene. We've crossed that to our neuropsin TET activator. And you can immediately see there's a real difference between how tau is distributed both in the cells and how it flows down into the projection areas, and how APP and A beta is distributed and where it goes to. This is a human antibody, and unfortunately this is meant to be A beta specific, but I don't think it is. This is APP, probably co-staining. Here you'll see we've actually aged these a little longer, and they've actually got amyloid plaques forming down there in the entorhinal cortex. So this is a pretty robust model with amyloid plaques forming there. Again, we'll be using these to compare and contrast our sort of two different pathological lesions. We can cross them together. We can turn off the pathologies, by use of tetracycline, etc. And we can actually see, we've already started to see some differences in distribution that might suggest interesting correlates in the human AD brain. One thing of particular note is the sparing of the dentate gyrus in this APP model. You usually don't see that spared in other mouse models, suggesting that this layer is actually very much affected by what goes on in the entorhinal cortex. And this is very similar to what you see in human AD. You see real sparing of the dentate gyrus in human AD and not usually. So I think this is actually very interesting in telling us that the entorhinal cortex perhaps is really a very fundamentally affected area for Alzheimer's disease. So I wanted to talk a little bit now about the mechanisms of tauopathy and how we're trying to intervene therapeutically. And this is a slide showing all of the different mechanisms that we can envisage going on. We don't know what initiates tauopathy other than when you've got mutations in tau, but we think it's probably some age-related degeneration of several systems. We have seen that elevated A beta can provoke and accelerate tauopathy. If you cross an APP mouse and a tau mouse, you get evoked pathology in those mice. And we're not sure, though, if it's a gain of dysfunction in that you've got something wrong with the tau aggregates, or if it's a loss of tau's normal function. And tau's normal function, as I said, is to stabilize microtubules. So you could have a significant disease effect if you lose its normal function, even if the tangles themselves are not important. It's something about loss of normal function. So here we have the hyperphosphorylated tau that forms because tau forms off the microtubules. It becomes a target for kinases and for phosphatases, and it can become hyperphosphorylated, accumulating into these aggregates that we see in the brain. This is some of the pathological events. Now, these are double-sided arrows. We don't know which one comes first. We don't know which is related to which. We do know the very first event is you see redistribution of tau from the axon to the somatodendritic compartments, and the very last event is cell death and dysfunction. Whether any of this is actually related to each other, we don't know. We're particularly interested in the role of hyperphosphorylation and the role of systems which are usually there to protect the cell from accumulating these aggregates. So we're interested in autophagy and the functioning of the UPS. So why are we interested in phosphorylation? Well, I told you tau phosphorylation is hyperphosphorylation. Abnormal phosphorylation is the very first one of the very first events we see. We see hyperphosphorylated tau in the AD brain, and this is lysate from an AD brain. And you can see here all the different phosphoforms with their different altered mobility when you run them on a gel. The tau protein is a highly phosphorylated protein. it's normally highly phosphorylated, and its phosphorylation status changes. So in the fetus, it has a certain pattern of phosphoepitopes that's different to the adult, and that's different again in the disease situation. In fact, the disease situation looks more like the fetal situation. But so that change in phosphorylation pattern is perhaps significant pathologically. We're very interested in whether an abnormality in phosphorylation or the kinases and the interplay between kinases and phosphatases actually can drive both plaque and tangle formation. And we know that aberrant phosphorylation is probably an issue in the aging brain. Here's a study on 3 months, 6 months, 12 months, 18-month-old normal mice with the different phosphoepitopes represented. You can see a significant shift in phospho-tau just in normal aging, which we believe in the human brain could actually set you up for some issues with aggregation of tau and A-beta. We know that the phosphorylation status changes with age, and also there's a regional differentiation. For example, there's very high expression of GSK and CDK5 just in the entorhinal cortex, and that could add a temporal and a spatial distribution aspect to this. Altered kinase activity, often associated with the lesions, some genetic linkage to some pathways related to especially GSK, but also some to CDK5. And to components of the GSK pathways, some of these are the Wnt signaling pathways, disheveled, catenin, LRP56, it's an LRP receptor protein family, and SORL1. So we've got a few things here that implicate kinases as potential players for that upstream dysfunction that I had pointed out on one of my earlier slides. Of all the kinases, GSK has received the most attention because it's been looked at. It has the best reagents to look at it, but also it's been implicated in all of these different ways. CDK5 has also been strongly implicated. So we wanted to look at two things. Firstly, the dogma that hyperphosphorylated tau is a pathological form of tau. We wanted to explore whether that was the case, or whether it was only a pathological form of tau in the context of a diseased brain. So we wanted to look at this in a model system that we developed. And this model system is based on hypothermia. When animals or when things become hypothermic, the kinases and phosphatases undergo a shift in their normal function, and you get a high degree of hyperphosphorylation of proteins, and tau is one of them. There are many proteins affected by this. This whole thermobiology aspect should be borne in mind, but we're looking at tau hyperphosphorylation specifically. And to induce hypothermia, we actually give the animals anesthetics. And there's two implications of anesthesia-derived hypothermia. One is, firstly, whenever you're doing experiments and you want to anesthetize your animals, just be aware they're probably dropping their body temperature and all sorts of things can happen to their phosphoproteins. And secondly, anesthesia has been implicated as a risk factor for Alzheimer's disease in elderly patients. There is some data that patients have a higher incidence of Alzheimer's disease several years after undergoing especially long surgeries such as hip replacements and cardiovascular effects. So we introduced our mouse models. We made them hypothermic by anesthesia, and then we looked at the phospho-tau. So here's three different anesthetics: chloroform, pentobarbital, isoflurane, and we identified that it was truly a hypothermia effect, because when you incubate the mice in a little oven and keep their temperatures normal at 37, the effect on phospho-tau goes away. And we looked at many different epitopes of tau and saw that they were all highly hyperphosphorylated. You see the shift in mobility when you look at total tau as the forms of tau go up to those higher mobility phosphoforms. Now, how tau is phosphorylated is an interplay between kinases that add phosphates and phosphatases that remove them. And what we saw was that in hypothermia, through a long series of experiments that I'm not going into detail here, we saw that actually the effect on phospho-tau and the effect of hypothermia was actually to inhibit phosphatase activity. So we have a model where we can induce hyperphosphorylation of tau, and then we wanted to see whether hyperphosphorylated tau maintained its ability to bind microtubules or not. The dogma has said that if tau becomes hyperphosphorylated, it loses its ability to bind microtubules, it falls off, its free concentration in the cytosol increases, it aggregates, and the rest is history. But we were concerned that maybe this wasn't the case. And so we looked at normothermic and hypothermic animals, and we did some fractionations where you could separate out the microtubule-bound tau in a pellet and the free tau in the cytosol. And essentially, even with significant hyperphosphorylation, we did not see any difference in the representativity of tau in those two fractions. There was some indication that the different phosphoepitopes of tau had different abilities to bind to microtubules, but overall there was no overt difference, even when tau was highly, highly hyperphosphorylated. Now, we did this in normal mice, and part of, we also looked at the effects on microtubules, on tubulin. So here were some tubulin blots, and here are the microtubules by EM. And we saw no overt difference in tubulin or in microtubule clusters, even at a fairly gross level by EM, but there was no overt difference, even in mice with that were hyperphosphorylated. So we thought, well, maybe in the normal situation, in a normal mouse, hyperphosphorylation is not what it's touted to be. It did not lead to overt tauopathy developing. And this was mimicked. This was made sense to us because there are models in nature, hibernating animals, where they become hypothermic for long periods of time, and they don't walk around with tauopathy at the end of it. So this made sense to us. And sure enough, this paper by Thomas Arandt's lab had shown that phospho-tau developed, but in fact it didn't go any further into a pathological form in hibernating animals. So we repeated these experiments in mice that had some tau pathology. This is the H-tau mouse line that I showed you the phenotype of earlier, progressive tauopathy, mild tauopathy. And we did similar experiments. We made them hypothermic. We increased the hyperphosphorylated tau amounts. And now we did start to see a significant increase in the free tau and a significant decrease in the pelleted microtubule-bound tau. And when we looked at the levels of different tau forms, this is total soluble tau, and this is the insoluble tau. You remember I told you there's this little 64 kilodalton form that seems to be associated with worsening pathology. We actually saw a shift to that higher molecular weight form of insoluble tau, suggesting they were worsened. In terms of pathology, there was a slight shift into the cell bodies in the anesthetized animals compared to the unanesthetized animals. And this isn't background staining, this is actually neuropil staining. This is phospho-tau that starts to pop up in the neuropil with anesthesia. These are perfectly controlled. But we did see a shift into the cell body, suggesting you were getting a more overt tangle formation. We were concerned here that maybe looking at them at the time of anesthesia was not relevant, because humans, they recover from their anesthesia and they show a worsening of their progression to Alzheimer's disease sometime later. So we took our mice and we looked at them 1 week after they'd been able to recover. We looked at them both at the time of anesthesia and also 1 week later versus control. And as you would expect, the level of phospho-tau at time of anesthesia was significantly elevated. The level of tau 1 week later had gone back to normal pretty much, with the exception of 88, which seemed to be maintained in a hyperphosphorylated state. What was particularly of interest to us, though, was that the level of insoluble tau not only didn't revert back to normal levels, it accelerated. It started to increase even 1 week later. And this was very worrying to us, because it suggested that, again, this self-propagating abnormal tau species can take hold and lead to more overt pathology later on. We saw a difference between males and females in our mice, and that probably reflects the level of pathology they have. There's a difference between male and female mice at any given age. The females required far less exposure to these hypothermic anesthetics than the male mice to progress, and it suggested, therefore, that it was tau-related, that you had increased the rate of acceleration if you had more incipient tauopathy at the beginning. In terms of the tangles, we didn't see an overt difference, and this is coming up again and again, that actually tangles, as measured by pathology measures, were not changed particularly. We're starting to really see a dissociation between pathologically measured tangle pathology and the stage of the disease. And this is happening in a lot of the neurodegenerative diseases where there's some other form probably that's actually important, and this could be the oligomeric forms, we don't know. But this pathology, the tangle load, doesn't necessarily reflect what's going on in the disease. So we were very, there's a number of implications of this. Obviously, anesthesia is something that patients undergo. Do they undergo hypothermia as part of their anesthesia, and especially in the brain, or is this just, you know, a mouse model thing? Well, the anesthesiologists tell us that yes, they do undergo hypothermia, and it's monitored but not corrected. So there is the potential that this is a situation in humans under long anesthetics that they would be exposed to conditions where this potentially could occur. Interestingly, tau is not the only thing to increase in its pathogenicity under anesthesia. APP processing is also evoked. BACE is upregulated, and there's more processing of APP under anesthesia. So potentially you could actually evoke both of these pathologies under anesthesia. So we wanted to really provide proof of concept that hyperphosphorylation, so that model is what, you can see all the caveats for a hyperphosphorylation induced by anesthesia and hypothermia. It's like, yeah, you know, that's so far removed from a real situation. But it allowed us to do a number of sort of fairly sophisticated things. The other model we had was a genetic model, and in this model we overexpressed the P25 activator of the kinase CDK5, and these mice have overactive CDK5 as they age, and they develop a little bit of phospho-tau in their neurons, not much, they don't develop tangles. When you cross them to a mutant mouse that has overt tangle pathology, and here's the sort of sarcosyl insoluble tau profile, you actually get a worsening, a considerable worsening of the phenotype in the double transgenics, both in terms of the biochemistry, you see the shift again to the higher molecular weight insoluble form, and in terms of tangle pathology. So this was really nice proof of concept. This was the first time that someone had shown that hyperphosphorylation of tau can be a pathological event. And the take-home message from all of this is that tau hyperphosphorylation is probably not pathological in a normal situation, but in a situation where there's some incipient disease, either in humans or in the mouse models, it is a factor leading to accelerated progression of the disease. Not only did we impact tau pathology, but we also actually found in those P25 mice with hyperactive CDK5 that actually A-beta levels were also increased. Here's A-beta 40 and 42 in the P25 versus non-transgenic mice. And we did a number of studies to look at all the processed fragments of APP, and it looked very much like APP processing was affected by overactive CDK5. To cut another long story short, we saw that this effect was due to an effect on base upregulation, and actually both the base mRNA and the base protein was increased in response to hyperactive CDK5. And we identified a mechanism through STAT3, which is a target of CDK5, that STAT3 was upregulated, and it upregulated transcription of base. So we actually have now two pathways that are affected by kinases. And again, I like this sort of thing because it fits in with our ideas that an upstream dysfunction, something general, maybe just aging or dysfunction of kinases, failure to regulate your kinase activity, can set off both pathways. And you might have to take care if you're going to just hit an A-beta pathway because the tangle pathway is still going on. So these fit our dual pathway type of mechanisms very nicely. In terms of therapeutics, all this work led on to a therapeutic test with a kinase inhibitor, and we'd done a lot of work with CDK5 and some with GSK, and there's a fairly nice GSK inhibitor out there, which is lithium, usually used for bipolar disorder, and several models, including our one, had shown that lithium GSK inhibition actually could correct the tauopathy in the mouse models. And lithium is now being tried in clinical trials for AD and PSP tauopathy patients. The downside of lithium, unfortunately, although it's FDA approved and it gets into the brain nicely, is that the patients don't tolerate it well, and so most of the patients come off the drug before the end of the clinical trial, and we're not going to have any data, I don't think, on effectiveness of this as a pathway. So, a lot of hope for phosphorylation pathways. Along a similar line, we were interested in cellular degradation pathways, and again, we're interested in removing more than one of the lesions in the brain, or affecting more than one of the pathways in a diseased brain. And the cellular degradation pathways, of which the UPS and the autophagy are two parts of the pathways, are very appealing because if you've got Alzheimer's disease where you've got plaques and tangles, about 40% of Alzheimer's patients also have Lewy bodies. And so now you've got three aggregated proteins in the same brain. And so we're very interested in pathways that will affect just the clearance of aggregated abnormal proteins as a therapeutic approach for having great utility. And so we're very interested in these pathways. Now, this is the UPS pathway, and the UPS pathway is usually a pathway for removing smaller aggregated proteins. Smaller aggregated proteins can fit into the bowel of the proteasome and be degraded. For larger aggregates and for organelles, another pathway is activated in cells, and that's autophagy pathways. Now, autophagy is there's a couple of different forms of autophagy, but we're interested in macroautophagy because it's been most implicated in the neurodegenerative diseases. So the signaling pathways that tell the UPS that it can't respond and signal the autophagy pathway to take over are pretty complicated, and I won't go into them in great detail. But we're looking at the interplay between these two pathways, both in terms of aging. Does this go down? Does this go down? Is this maybe why you get these lesions forming? But we have more interest in autophagy as a possible therapeutic target for the neurodegenerative diseases. And so this is the autophagy pathway. There's a couple of different ways to approach it, but most of it is mTOR dependent. And what you don't need to know all of the details of this, but the pathway leads to the formation of a particular vesicle, a double membranous vesicle, which forms in response to signaling, and it's called an autophagosome. And it's recognizable because of this marker here, the production of LC32, which really is a good marker of the formation of this autophagic vacuole known as an AV, which then goes on to fuse with a lysosome, leading to degradation of the contents of the autophagosome, which can be altered abnormal mitochondria, aggregated proteins, etc. So this whole system is a way of identifying and packaging up abnormal proteins and organelles for delivery to the lysosome where they're finally degraded. And you can see that autophagic vacuoles are very distinctive. They tend to accumulate in the brains of abnormal-looking neurites in the brains of patients with degenerative disease and in the mouse models. And there's a number of tools for looking at them. Here are LC3 GFP-labeled puncta, showing that autophagic vacuoles have been formed. So we look at a number of markers when we're trying to identify whether this pathway has been activated. We have a number of drugs to intervene here. Rapamycin is the classically used one, but we have a number of others, and I'll show you a piece of data with that. So what we're thinking is happening in the tauopathies is autophagy can be induced, but it has to go to completion to actually degrade the contents of the AVs and bring them into communication with the lysosomes for degradation. And a lot of that's about transport. These AVs are transported up the axon, and here's normal transport, but here's transport that's blocked, perhaps by a deficit in axonal transport, and you get accumulation of the AVs. They're induced okay, but they can't go to their ultimate destination. So we're very interested in the effects of axonal transport and the accumulation of AVs and the failure to complete autophagy. So one of the first things we wanted to do is we looked in our mouse model of tauopathy. This is the inducible mouse model of tauopathy. And we wanted to look at a number of measures to give us some feedback about the status of autophagy in these mice as the pathology worsened. And here's the worsening pathology with 3 months, 4 months, 6 months, 8 months of age. Here's the appearance of that insoluble form of tau at 64 kilodaltons, comes in about 4 months of age. Here's some abnormal phosphoepitopes. Ubiquitin I should have on here the non-transgenic or even the 1-month-old. Ubiquitin is highly represented in these mice and exceptionally so after 8 months. The whole UPS system seems to completely crash as the mice get older. But interestingly, autophagy is also upregulated. Here's Becklin, one of the first parts of the pathway. It's upregulated as the pathology increases. LC32 is upregulated here also around the same time. And so we're doing some mapping studies to look and see when the UPS shows dysfunction versus when is autophagy upregulated, and then does it go to completion. And this is the situation in humans. This is a frontal temporal lobe dementia and a PSP brain. And you see essentially the same thing. P62 is a particularly interesting molecule because it's an agrosome. This usually goes with the aggregates that you see forming in the brain and their removal. So you see a similar situation in the ubiquitin protein, insoluble tau, markers of autophagy are upregulated with worsening pathology. And sure enough, when you look at the AVs by ultrastructure in the mice, you see an accumulation in the axons. So we've done a lot of work with this, but I think this is the most important bit, is that we've been using some therapeutics to try and correct what we believe is an autophagy-based problem in the mice. And there are two drugs. I mentioned rapamycin. Rapamycin is not brain penetrant. So we wanted to use a drug that is brain penetrant, and that's trailose, which is a simple disaccharide, which induces the beginning of autophagy. And we took our mouse model, and our mouse model has motor impairment. And this is yet another line of tau mice, the JMPL3 mice. And sure enough, we could, with trailose administration, we could reduce the levels of insoluble tau, especially that higher molecular weight form. We could improve their measures on a motor test, and we could improve their measures in a rotor rod. And we have a lot more data on this, but I don't have time to go through it in detail. Suffice to say, autophagy does seem to be a good pathway to intervene, and we're doing some studies now to see if it corrects cognitive impairment. It's not a new idea. Autophagy has been implicated in Huntington's disease. It's been looked at in Parkinson's disease, and I think that's all good. I think if we can induce autophagy and actually do something with the pathological lesions in any of these diseases, especially if they co-occur in the same brain, then that's a good thing. So just to end up, I just wanted to show you a new system that we're working up to look at transport. You remember I said that I think a lot of this is about transport deficits related to the transport of autophagic vacuoles and the buildup of AVs and failure to complete. Well, the model system we're looking at is a microfluidic system. And what you do in a microfluidic system is you take primary cultured neurons and you have these chambers, which are, it's really one set of chambers and a second set of chambers, and they're linked by these microgrooves. And these microgrooves are shown here. And what you do is you grow your primary cultures on one side of the chambers here, and the axons grow through these microgrooves and form a dense network on the other side. No cell bodies on this side, just a dense network of axons. And these microgrooves are of sufficient length that only the axons grow. So this is an axonal marker. This is a dendritic marker. And you can see, depending on the length of the microgrooves, only the axons can actually get through to the other side. So you kind of screen out the dendrites. And this is a really nice system for first linearizing a cell system. Only one or a small number of axons actually grow through these grooves, so you can really look at individual axons. And that's shown here, and this was a movie, but it's not working here. This is a single axon, and this is stained with mitotracker. So some of the cell bodies are stained with mitotracker, and you'll see the movement of these mitochondria. Here's one with several axons, here's one with just a couple, and here's one with one. And by monitoring the movement of organelles, and we're doing mitotracker here, but you could do LISA tracker, or you could do LC3 labeled AVs, GFP labeled AVs, or any of your organelle of choice that you can label. You can actually monitor the movement, and here is one moving anterograde, here's one moving retrograde, and here's one that just stayed the same and wobbled about in position. So these are really nice systems, and I kind of just am showing this to you to bring it to your attention, because these microfluidic systems are great for studying transport. And also for looking at what happens in axons versus cell bodies. You can actually really separate things out really nicely and apply drugs or look at distribution of proteins and things. But what we saw in these, and this is the beginning of a study that we're going to do with rapamycin or trailose, what we were able to see was that the movement, anterograde movement of mitochondria in our primary cultures derived from our transgenic mouse. And this primary culture develops insoluble tau. It's unusual for tauopathy to have a cell model that develops insoluble tau. This is an insoluble tau developing cell model versus a non-transgenic. And what you can see is that the transport of mitochondria is already seen to be deficient in those mice. And here's the bins. You've got more movement at the slower time frames and more movement in the higher ones for the non-transgenics. So I think this system's going to be really nice for actually looking at the effects of drugs that affect axonal transport. So as an overview, Tangle pathology correlates best with cognitive decline in AD, not amyloid plaques, and it doesn't seem to be prevented in patients with reduced amyloid load following immunotherapy. So I think therapeutics against tau, and that could be tangles or the precursors of tangles, are likely to be necessary to reduce cognitive decline and degeneration once the disease has started. And this is a really important point. Once the disease has started, this is what we can't do at the moment. We can't get ahead of the disease at this point. The disease is likely to be well advanced even at the earliest stages diagnosed, which is MCI or mild cognitive impairment stage. And so I think it's really advantageous for us to develop therapeutic approaches that target more than one pathological entity, plaques, tangles, and Lewy bodies. And these may be more efficacious for diseases such as AD that have mixed pathology. We also need to invest more in tau therapeutics because I think we're going to be left with a tauopathy from Alzheimer's disease if you just target the A-beta part of it. And so that's my message to NIH, is that I think we need to really think more about the impact of tangles, even though the genetics doesn't implicate it as a primary lesion. I think it is a primary problem in Alzheimer's disease that we need to target therapeutically. So thank you to my lab, my ever-dwindling lab. I have a couple of people, one person working on the microfluidics, Ho Yu is working on the autophagy, Li Lu is doing the mouse models for the spreader pathology, and my postdoc is looking at methylene blue as a therapeutic, you might be interested to know. And I have several people who contributed to the work shown here who've moved on to bigger and better things. So thank you very much for your time. I have no clock, so I have no idea what time it is. Yes?
Excellent work and presentation. So I thought it might be much easier to deal with the extracellular amyloid insoluble proteins rather than the intracellular tau targets. So when the antibody was used, did anybody look for the real clearance from the brain into CSF or into blood? Do you see an increase of the complex and antigen, which is amyloid beta, with the antibody in the blood?
Right. So is that several parts of a question there? Was it increased in the CSF? Are you asking, or well, what they think happens is that it actually does drain into the CSF. The plaques are resolved and it drains the CSF. What you do see, unfortunately, with the immunotherapy is an increase on the blood vessel walls. So you might actually be stimulating angiopathy as you're removing parenchymal amyloid. So that's a bit of an issue. In terms of what got into the CSF, it's not been monitored with these patients, but it will be monitored in the future. The CSF is a little difficult because the levels of CSF A-beta decline as pathology increases. As it gets sucked onto the plaques, it's less represented in the CSF. So we don't know whether it will increase or decrease in the CSF, but it's something people are monitoring. They're not sure what to make of it when they get it. In terms of the plasma, the CSF should drain to the plasma. The antibody binds A-beta in the plasma and increases its concentration 1,000-fold or so as it binds A-beta. And we're not sure if that's sucking it down from the brain or if it's just preventing peripherally produced A-beta entering the brain. That's the peripheral sink idea, but we don't know what's going to happen to the relative levels of A-beta in CSF, interstitial fluid, plasma. It's coming. All those data are coming. They're doing new studies now with bapamuzumab, which is an antibody-based immunotherapy. So, is that okay?
So in terms of clearance, unlike the rest of the body, brain is almost a closed system. So any of these approaches then might be, I guess, sort of inhibited in terms of clearance pattern, although you're making all this similar approach which works in the rest of the body. In the brain, because of the difference of access to through the blood-brain barrier as well as other inhibitions, this might be a real challenge. The
antibodies get into the brain, and the A-beta seems to get out of the brain. So, although it's a closed system, there's some fluidity there. Also, don't forget, a lot of Alzheimer's patients have compromised blood-brain barrier, so that might actually open up a lot more drainage out of the brain. But we're not sure where it goes to with the immunotherapy. But it seems to get out of the brain. It certainly it's not in the parenchyma very much. It is on the blood vessel walls to some extent, so that might be an issue. But yeah, it's a risky therapy, but it's the only one we've got that actually reduces amyloid in the brain in a human for us to monitor what's going on. They are developing vaccine therapies with tau, which is even more amazing, because that has to not only get into the brain but into a cell to actually target a lesion. And in the mouse models, immunotherapy for tau has actually been quite successful, for phospho-tau. It's also been done for synucleinopathies. So this whole immunotherapy might actually be quite an effective therapeutic.
Sorry, how do you target the autophagy pathway to taking up just tau? Or do you just non-specifically just rev it up? And
no, just rev it up. I mean, we've just got tau mouse models, but no, it should hit, anything. I think it'll only hit the abnormal. What we're hoping is, as I showed you in that time course, with worsening progression, autophagy is induced. So I'm hoping that you've got an abnormal situation, so it shouldn't hit normal. In a normal situation, it shouldn't have any effect. Really, more so in an abnormal situation. So. But it should hit any protein that's aggregated, which I think is part of its beauty.
Karen, so for the last part of your presentation, you talk about the microfluid chambers. And have you seen any kind of change of the growth of axons in your transgenic neurons?
That's an interesting question. We thought we would. We thought we would see a reduced outgrowth of the axons. We haven't seen it particularly. It's not particularly strong if it's there. They seem to grow about the same rate. Now those cells, they they progressively accumulate insoluble tau. So we look at them at around 10 to 14 days in vitro, and it's possible that they've done most of their growing before the insoluble tau has actually taken and it had an effect on outgrowth. But over the rate at which they get through those microgrooves is not overtly different.
Okay. So besides you track the mitochondrial transport, and have you checked another organelle? So some I think it's probably is.
We're looking at LISA tracker. We're using them also to look at transfer of molecules between cells. So one of our main interests, and I can't show that it's actually here. I can't show the data because it's really, I don't quite understand it yet. But we're growing cultures from a transgenic on one side, a tau knockout on the other side. One of them has got GFP, the other one is labeled with tau. And what we're looking at is as the axons pass, they come out both sides. And we're looking at the tau negative cells, see if the tau is transferred into them by confocal, if there's a merging of tau and GFP, which would suggest the physical transfer of tau from a tau positive cell to a tau negative cell. And so they're very nice for that sort of thing because it's linear and you can grow, different cultures of cells. You can actually have 4 sets of chambers. You can have all sorts of things growing together and putting their axons onto cell bodies and things.
Yeah, it's very interesting.
Yeah, they love. I kind of that's my kind of little interest. I know I haven't got much data on it yet, but I thought it might be worth people knowing that these things are out there and usable for studies that we're interested in. Are you coming to pull me off?
Just wanted to thank again Dr. Duff for coming and giving this interesting lecture, and I hope you can join her and at the reception in the library. Thank you.